Okay, let's start with a quote. The more you know about the past, the better prepared you are for the future. Okay, let's begin the class. So today we'll see about history of microbiology. So before moving to the history, let's make a quick review of microbiology. So microbiology is the study of microorganisms which is visible only with a microscope. For example, bacteria, virus, protozoa, etc. So most of the microorganisms are harmless. Only 1% of microorganisms makes complication to us. Okay, history of microbiology is made divisions into a discovery era, transition era, golden era, and modern era. Let's see one by one. Discovery era. This particular two scientists, Aristotle and Antony von Leeuwenhoek, completely believe in the spontaneous generation. So, spontaneous generation is nothing but they believe that living organisms could develop from non living materials. So, these are the specific particular scientists have seen the first microorganisms in the on the living non living materials. So, Antony von Leeuwenhoek he first made a microscope. With the help of that microscope, he used to see some tiny living organisms is moving on the non-living materials. Then he started to see about that particular microorganisms that move, and he used to make a structure of specific living organisms. Okay, some may be rod shape, some may be spiral shape, some may be a bundle of tuft-like structures, grape tuft-like structures. He made all the pictures uh, of microorganisms. In the 13th century, Rogan Bacon described that the disease caused by a minute seed or germ. He they started to they started thinking about the microorganisms of the movements, and he specifically this Rogan Bacon described the diseases caused by these specifically microorganisms. Okay, and Antony von Leeuwenhoek is the father of bacteriology, protozoology, and of course microbiology. Father of microbiology is Antony von Leeuwenhoek. Okay, let's Antony von Leeuwenhoek he used to call microorganisms as the animalcules. So the animalcules is nothing but the tiny living organisms which is present in the world. Okay, let's see the foundations, the contributions of Antony von Leeuwenhoek. He built a simple microscope with only one lens to examine the blood, yeast, insects, and many other tiny objects. See this microscope, how it is there. How you will see in the laboratory, that is one compound microscope. This is a simple microscope. See, with a one lens he made. With the one lens he used to see the microorganism movements. So he invented the first practical microscope and used them to become the first person to see and describe bacteria among other microscopic discoveries. He discovered blood cells, microscopic nematodes, and studied the structure of wood and crystals. Okay, he also made over 500 microscopes to view specific objects. Okay, he became an expert on animal reproduction. He used to see the tiny microorganism reproductions and everything. Okay, see, this is about the transition era. See, the particular scientist like Francisco Redi, he showed that maggots would not arise from decaying meat. Okay, maggots is nothing but you can see this picture here. Maggots is nothing but these are the legless larva which is found in the decaying meat. He completely showed that maggots would not arise from decaying meat. Okay, because of the microorganisms, he started to prove that because of the specific microorganisms, maggots will develop. Okay, and this John Needham, he is the supporter of spontaneous generation theory, and he proposed that tiny organisms arouse spontaneously on the mutton gravy. They have seen first in the mutton gravy the specific microorganisms is present. He has seen by using the specific microscopes in the mutton broth or mutton gravy. Okay, after the contamination, you can observe, right, these microorganisms like that. They have seen the first. John Needham and Francisco Reddy specifically showed that maggots would not arise from the decaying meat. And this Lazaro, he demonstrated that air carried germs to the culture medium. So we prepare the culture medium, right? In the air also we know that today we know that microorganisms will present in atmosphere. At the time, he specifically this Lazaro, he demonstrated that air carried some specific germs to the culture medium. He showed that boiled broth would not give rise to microscopic forms of life. Okay, every specific microbiologist or any scientist has made a contribution in this transition era. Okay, these are the specific scientists: Francisco, John Needham, Lazaro Spallanzia. In golden era, specifically this Louis Pasteur, he's the father of medical microbiology. He made a technique of a pasteurization. So if we boil for, if we heat for 1 to 2 minutes or 3 minutes, specifically for he made for 30 minutes, all the microorganisms which is present in the food item will die. He proved that thing, that specific technique is nothing but the pasteurization. Okay, he made a experiment in 1897. Mild heating at 62.8 degrees centigrade for 30 minutes, boiling was enough to destroy the undesirable microorganisms which is present in the. It won't affect the taste of product. So this technique is nothing but pasteurization. How we will boil for 3 to 4 minutes of milk. So whenever we will take the milk from the shops, we will boil, right? Why we are boiling? To destroy the undesirable microorganisms which is present in the milk. So that is nothing but the pasteurization technique. He developed this. Louis Pasteur has developed. Louis Pasteur have made lots of contribution to the microbiology. Let's see one by one. So he invented the process of pasteurization, as I told, and he also invented the process of fermentation. Okay, how the milk will convert to the curd with the help of specific microorganisms. Okay, he developed that thing, he invented. And development of effective vaccines like rabies vaccines and anthrax vaccines. So rabies vaccine. So rabies is one of the deadly virus which is present, which will carry by the infected animal, or it may be present in the saliva of specific animal. Specifically, we can see in the dogs. The dog saliva, you can see the rabies virus. That particular rabies is will complicate to us. So anthrax is one of the bacteria which will present in the infected animals. Okay, and this Louis Pasteur has demonstrated diseases of silkworm was due to a protozoan parasite. So because of this specific protozoa, he demonstrated the silkworm disease, and he made lots of contributions. He coined the term microbiology. So we are listening the microbiology, right? He has given that name. So we are listening aerobic, anaerobic. What is that? He made that particular words by using the Latin and Greek words. Greek language, he made aerobic, anaerobic microbiology words. And he completely disapproved the theory of spontaneous generation. Okay, spontaneous generation is nothing but the living organisms which come from the non-living materials. He completely disapproved that particular theory. And he demonstrated the anthrax was caused by specific bacteria and also produced a vaccine for the disease. Okay, he developed the live attenuated vaccine for the disease. So live attenuated vaccines will destroy the or it will destroy the virulence of pathogens which makes the disease which develop the disease in our body. The process will shut off. And lots of contributions. He suggested that the disease free caterpillars can eliminate the disease. So he isolated the anthrax causing bacilli from bloods of cattle, sheep, and human beings. He demonstrated the virulence, which having a capability to cause disease. And he demonstrated the principle of immunization. It is very important. Immunization concept has developed by Louis Pasteur. He demonstrated, and he called attenuated cultures as a vaccine. And he developed lots of vaccines against rabbits from the brain and spinal cord of rabbit. See, he made lots of contribution towards the microbiology. So heating the juice for 30 minutes will kill microorganisms. So as we told, this technique is known as pasteurization, and he discovered the lots of parasites causing pebrine disease of silkworm. So we discussed in the last slides. And he developed a method to remove the microorganism from juice without affecting the quality. How? By heating the juice. And specifically, he's a chemistry professor in the France, but he made lots of contribution in the microbiology. Okay, so fermentation process. He completely disapproved the spontaneous generation, and he's got a different microbes giving different tastes of wines. So how the wine, uh, how this wine will manufacture with the help of some grapes and with vitalizing the microorganisms in under the earth. With the help of the microorganisms, wine will develop, wine will manufacture. Okay, so that is about the Louis Pasteur contribution. Let's see some other scientist, John Tindall. He discovered highly resistant bacterial structure that is present known as endospore bacteriums. Okay, he prolonged boiling of intermittent heating was necessary to kill these spores to make the infusion completely sterilized, a process known as Tyndallization. Okay, the process of Tyndallization has developed by John Tyndall. Okay, and we'll see about Joseph Lister. He is the father of antiseptic surgery. So Lister concluded that wound infections too and were due to microorganisms. So wound infections will happen due to specific microorganism. He concluded that thing. Okay. Then he also devised a method to destroy microorganisms in the operation theater by spraying a fine mist of carbolic acid into the air. Okay, now we are using some specific sprays in the operation theater to destroy the microorganisms. This Lord Joseph Lister has developed that specific spray. Okay. Then Robert Koch is one of the a particular scientist. He also made some lots of contribution. So he demonstrated the role of bacteria in causing disease. He demonstrated that thing, Robert Koch. And the technique of isolation. So we have discussed about pure culture techniques, right? All these isolation of pure culture techniques has developed by this Robert Koch. Okay, Robert Koch used gelatin to prepare culture media, but it was not an ideal because gelatin is a protein. It is digested by many bacteria capable of producing a proteolytic exo enzymes. Because gelatin is used as a gel-like form. Because of gelatin is a protein, bacteria will utilize that protein for the cell growth. Okay, because of this limitation or because of this limitation, uh, at present they are using, instead of gelatin they will use as a agar. We have seen in the raw materials of culture media, right? Agar, this particular agar, after getting solidified, agar will it is not a food for the microorganisms. It will make a platform for the microbial growth. Okay, see, Robert Koch has made some contributions. See, so the same microorganisms are present in every case of disease. So because of the specific microorganism, the particular animal has died, and he demonstrated and proved that specific process. See, he has taken the microorganisms are isolated from the tissues of a dead animal, and a pure culture is prepared here. Right here they have prepared, and that pure culture as injected to the healthy animal. See, microorganisms from the pure culture are inoculated into a healthy, susceptible animal. The disease is produced. Then the particular animal has died. And he has taken from the dead animal, and again he isolated a specific microorganism, and he proved that because of this specific microorganisms, that particular animal has died. So he made this contribution. He has proved to the world. So specific microorganisms will kill the specific animals. Then this Robert Koch has discovered the anthrax disease cycle and the bacteria responsible for tuberculosis and cholera. So we know that Mycobacterium tuberculi is making complications. If the particular microorganism has entered in your body, make some lung infection, that particular disease is known as tuberculosis. We know that, but he discovered that thing. Okay, Robert Koch, and he made a concept of spear making and staining with aniline dyes, and he got a Nobel Prize for physiology or medicine, specifically in 1905. And he made the concept of cultivation of bacteria on solid media and hanging drop method. Robert Koch has developed this hanging drop method. um, if you see in hanging drop method, we can see the motility of bacteria. We can see the microorganisms moving with the help of the compound microscope. Okay, and he also discovered the causative agents of tuberculosis, cholera, and pink eye. And he developed the methods of testing disinfectants. These are all contributions of Robert Koch, and he got the Nobel Prize for his work on TB in 1905. That is about the Robert Koch. And she is the assistant for Robert Koch. She is Fan Alchemius. So she is the assistant to the Robert Koch. She proposed the use of agar in culture media. Okay, instead of gelatin, we can use agar in culture media for bacterial growth. Why? Because agar is not attacked by the most bacteria. So agar is better than gelatin because of its high melting point and solidifying agent. Okay, this is the main thing she has developed. Then Richard Petry. He developed the Petri dish plates, what we are seeing in the laboratories. Petri plates will call, right? This Richard Petry, he developed the Petri plates, a container used for solid culture media. Okay. And Edward Jenner, first to prevent the smallpox, he discovered the technique of vaccination. And Alexander Fleming, he discovered the penicillin, as you know very well, penicillin from the Penicillium notatum bacteria that destroys the several pathogenic bacteria. And Paul Ehrlich, he discovered the treatment of syphilis by using arsenic. So it is a sexual contaminated disease, syphilis. He discovered the treatment for the syphilis, and he also studied some toxins and antitoxins in quantitative terms and laid foundations of biological standardization. And he also studied this. Paul Eldridge has studied the lots of toxic substances and antitoxic substances. Okay, and important discovery. See the bacteria, this particular Hansen from leprosy, bacillus. Okay, this Neisser. Lots of scientists have discovered. See, in a specific year, these are the viruses who coined the term virus for the filterable infectious agent. He's Bizarink. Okay. So vaccination for smallpox, Edward Jenner, we have discussed in the last slides, bacteriophages, Chott and G. Harrell. These are the specific scientists made a contribution to the microbiologies. Okay, Louis Pasteur developed the rabies vaccine. So coming to the modern era, see, I made a tabular column. In that particular year, they got Nobel Prize for the contribution. So Robert Koch, we have discussed the Malay tuberculosis. Ronald Ross contribute to the malaria for the treatment and that particular malaria cycles and everything. He has given contribution to the particular disease. See, prisoner to the prions. Did you remember the prions? It is one of the infectious agents which having a capable to make the disease. So prions are misfolded proteins that are capable of making some neurodegenerative diseases, specifically Parkinson's disease. Okay. So this Brenner and Harvard's a genetic regulation of organ development and cell death. Okay, lots of scientists made contribution to the microbiologies. Okay, this is about the history of microbiology. Thank you. Have a nice time.